Fiona McRonald and I are going to do a double act in this, but before we do, I had to just tell you that something weird happened to our family. 2 years ago, our grandchildren and our daughter and son-in-law sang a song for Linda's birthday and put it on YouTube and went out to walk the dog, called One Day More, Rewrite for COVID. And when they got back from walking the dog, it had been viewed 1 million times. And a year ago, they were actually on Comic Relief with their subsequent blockbuster, and they've now got about 30 online. And in November, they made one for Prostate Cancer UK, which we are featuring to celebrate the 3rd year since I had my prostate removed for prostate cancer. And it's called Back the Knife, and it's very entertaining, so I would encourage you to watch it. We even made the local paper, Newcastle Hospital Boss makes cameo in prostate. So officially, thank you. I'm very famous in Lynch syndrome circles, but actually I am officially the 8th most famous member of our family now. The grandchildren, daughter, son-in-law, and their dog have more followers than I do. Anyway, so this is our talk, and Fiona and I, Dr. Fiona McRonald, who works for the National Disease Registration Service, who is currently gossiping with Ian, but she will be joining me shortly. I'm going to give you a quick update on aspirin, because we've already talked about it. We're going to talk about the national screening progress for bowel cancer, which Fiona will present, and then I want to show you some very exciting new stuff before we go and have a beer. So first of all, we've talked about this. You saw that very nice, Andrew showed the slide, but this is the paper that came out in 2020, which was the official 10-year follow-up of our CAP2 study, which many of you took part in. And the important take-home message here is, first of all, that graph, that's what's called a cumulative incidence. So basically the red line, the people who were blindly taking aspirin had half as many cancers, and these are the people who actually took the aspirin. It was a 50% reduction in the number of colon cancers. The people who were on the placebo tablets, by about 15 years out, were up to 30% having had colon cancer. So actually, officially, aspirin prevents more colon cancers than colonoscopy, which is an interesting take-home message. And that's going back to what Ian was saying, is that we get these cancers just popping up. And so they arrive as cancers, which was always assumed that colonoscopy would prevent cancer by taking the polyps out. But if they didn't come into polyp stage, then we just don't see them until they're there. So when someone says you shouldn't take aspirin, that's the answer. And in fact, the risk of the colonoscopy is higher than the risk of the aspirin. If you actually tot up the number of times you get bleeds and various, I mean, they're rare, but they do happen. Okay. So the other thing is that if you actually look at this the other way around, and we did the left-hand side of that graph is the official 10-year follow-up, but we got permission to, with the help of Jem, who is the boss over Fiona's team, we managed to get access to all of the data on all of the people in Britain who've taken part, because all of our cancers are notified to the National Disease Registration Service, so we could actually track people even if we'd lost them, because the ethicists agreed that if they'd agreed to be followed for 10 years, well, it's okay to have another look at the next 10 years. And so that gave us that very powerful data. And the important thing is that none of us knew who was on what until about 10 years out. So this is truly blind data. And as a result, NICE recommended that we should all take aspirin if we've got Lynch syndrome. And if you are having a problem with your GPs, we heard about from Kelly's project earlier, which is funded by this 5-year money from Cancer Research UK that we got. You can go look at that website, nice.uk, nice.org.uk. Just put in aspirin, and the thing that comes up at the top is Lynch syndrome treatment. And we helped them write a very nice guidance, which you can print out and take to your doctor and say, have a read of that. CAP3, one of the criticisms is, well, we don't know what dose to use. Well, we don't know how often to do colonoscopy, but we still do it. And so similarly, we don't quite know what the best dose of aspirin is, but some's got to be better than none. But the trial, which Jill is central to keeping alive and has done such a fantastic job with over several years, is now reaching its conclusion. We've got a couple of years left to go. So we randomized 1,879 people with Lynch syndrome. Some of you in the audience are in the study. And we did it blind at first, so we could see what the true side effect profile was when you didn't know how much aspirin you were on. We were going to stop the aspirin then, but we redesigned it in the course of the study because we were hoping to get slightly more people in than we did. And to give it more power, we've kept going on open label, because since we're looking up on the national database for the cancers, it doesn't matter if you know what dose you're on, because we're actually counting the cancers objectively. So we'll have the answer to that in 2024 and probably have the result. The last patient, last visit will be 2024. I've got a whole bunch of samples we're collecting and doing other work, which we will talk about at other times. And Nick is now out the end. He's done his 5 years, as many of you have. And so what I'm saying is that if you want to know what I'm suggesting is the sensible doses, if you're under 70 kilograms, 11 stone, 10, 11 stone, take a half an aspirin a day. If you're over that weight, take a whole aspirin. And there's lots of science behind that. The other thing is that CAP2 was actually a factorial trial. We did 2 interventions, and many of you who took part will remember the pleasant joy of taking powdered starch every day for years. And so what we actually did was, this is now finally going to be published. We've had real battles with the editors of journals and their referees, because we were expecting the starch to reduce the burden of bowel cancer, and it didn't at all. But when we did the final analysis, we said, let's just have one more run past the starch. And to our amazement, we saw an enormously powerful effect. But the effect wasn't on the Lynch syndrome colon cancers, which is what we as our primary endpoint. It was the other cancers that had gone down. But as you can see there, with a p-value of 0.005, the incident rate ratio, that's if you that counts all of your cancers, was down at 0.38. So there's a 60% reduction in the number of other cancers. And when we drilled down on that, you can see the orange box there. It's the upper GI cancers, which were significantly reduced. And so this will be coming out in cancer prevention research. We're just responding to referees now, but this time we're going to get it published. And so I think the exciting thing about this is that this is a simple dietary modification. I can give you some theories as to why it might be working, but to be honest, I don't care, because it's just a question of having a high fibre diet, which isn't hard to do. And I endlessly used to talk about slightly green bananas. So a slightly green big banana, you know, the green tipped ones, so just a bit firm, will have about 30 grams of resistant starch in it. So as it softens, it turns into sugar. But while it's still a bit bitter, it's got lots of resistant starch. But you can also get it from lots of other foodstuffs, and we can do a daytime television program about it maybe. Anyway, I should say that just one banana a day, we did many years ago first start doing this, and one of our patients' wives said, he's a long distance lorry driver, and she said, can you tell him to stop drinking bananas? He's up to 23 a day, and he's leaving the skins in bed. Anyway, more seriously, what about the adverse effects of aspirin? So this is the graph that Mangesh and Jack produced, our partners in ASCAP. So basically, what you can see that by the time you get to 80 to 84, it's about 1 per 1,000 patient years having fatal GI bleeds. So this is kind of serious stuff. It's still a very small number, but if you're treating thousands of people, that's an unacceptably high death rate from bleeding. Now, the important thing is that many people put on aspirin when they're in the 50s and 60s, and then nobody bothered to take them off. So they're still at 85 taking aspirin, and they shouldn't be. And as we said earlier, we should be stopping the aspirin as we get older because our guts can't tolerate it. But there was a big study set up by John McNeill and colleagues in Australia, which was unfortunately, we kind of could have warned them, because if you look back at the history, in Paganini Hill's study in 1995, they looked at really old people in old care homes, and the aspirin didn't prevent colon cancer in that group. But they nevertheless soldiered on, and they recruited thousands of people and started putting them on aspirin from the age of 70. And so far we haven't got to the 5-year mark yet, so we don't know how many cancers they've prevented, but they caused a shed load of side effects. So this has caused a huge anti-aspirin movement, and it's mostly because GPs and physicians look after old people. You know, most of the people in hospital are over 70. I know, I've been there. So you know, we should just hold our line and say for Lynch syndrome, the side effects are minimal in people in their 40 to 65 age bracket. If we can get you on 4 or 5 years worth of aspirin in that window, you can safely stop the aspirin, because you'll see, as you saw from the graph, that benefit extends out for about 15 years. I also just want to quickly show you that everybody likes cartoons. I love this cartoon. My friend Chris Heinen gave this talk, and it's a 30-minute talk. It's like the London to Brighton in 3 minutes. Remember that old thing where they speeded it up? So I've speeded up his talk. So this is a mismatch where you should have had a C, but you've got a T, and the DNA doesn't stick together when the copy's being made. So here is how these proteins work. and I've just sort of speeded it up. So essentially you get the MSH2 and MSH6 form little clamps, and they run up to where the replication's got to, fill it up all the way back, then MLH1 and PMS2 arrive to activate it, and then XO1 comes and strips off all the bit that you've just made, and it puts that purple thing on. I've forgotten the name of it. It doesn't really matter, because the point is it then brings back your polymerase and runs it again, and it fixes it. So essentially that's how these proteins, and as I said that was happening in one of your cells at least, in everybody in the room. In every living thing, this system is essential. It actually is the spell checker on copying DNA for all living things apart from viruses. We don't count them. So I'll now hand over to Fiona, who Fiona and I and Jill and Stephen Hardy and colleagues have been doing this fantastic study together. And Fiona's now going to tell you, this is started off with a grant we got from Bowel Cancer UK and is now carried on by Claire Turnbull's money. I'm just holding it, it's easier than...
Hello. Well, I was going to start by asking who has heard of the National Cancer Registry, but John's going to mention it, so now everyone's heard of it. But who was aware of the Cancer Registry before John mentioned it today?
Well, quite a lot.
Yeah, about half the people in the audience. Okay. Well, I'll tell you a little bit about us, because not everyone does know about us. So the National Cancer Registration and Analysis Service, NCRAS. It's one half of the National Disease Registration Service, and our other arm, which is a bit newer, registers congenital anomalies and rare diseases. So in the case of Lynch syndrome, that fits very nicely into sort of both categories. Now we have a statutory function to collect patient data from anyone diagnosed with or treated for cancer on the NHS. And we've got a legal statute that enables us to do this without explicit patient consent. And the reason is because our work is in the public interest, and it's a vital part of public health work. So if you go on the Cancer Research UK website, you look at the national cancer statistics, you want to know how many people have had bowel cancer, what the survival rate is. You can find that out from the Cancer Research UK website. Where do those statistics come from? They come from us in the National Cancer Registry. We do the official national cancer statistics. And we were part of Public Health England, but we moved over to NHS Digital in October last year. So organisational change is always delightful. This is who we are. We're a slightly bigger team than this, but this is just the people who are working specifically on Lynch syndrome. We all work on other cancers as well, but we're involved in the Lynch work. So Stephen leads the team. He's a lovely Geordie like John.
He's actually a Mackham, but we all voted against him.
Oh, okay. Yeah, true. He's a Mackham, but he is lovely.
Yeah, he's lovely. He is lovely, apart from being a Mackham.
Apart from being a Mackham, yeah. At least he's a northerner. And then there's me there, so we're kind of leading and managing the team. Our analyst in the middle is Jo, so she's done lots of the number crunching for this, and Francesco and Ollie do all the really, really clever stuff with the computer code. So it's quite multidisciplinary. So as a cancer registry, this is broadly how we work. So the circles at the top show the sort of story of somebody with cancer. You start off with a genetic risk, and the rectangles at the bottom are all the data sources that we collect into the registry that relate to all of those. So there's the patient journey and all the data sources. So for us in the molecular and genomics team, the data sources that we are interested in are the genetic risk data, so that's the germline mutations that most of us in this room have got. And we also collect the molecular data, which is on the tumours themselves, and I'll talk a bit more about that later. At the other end of the scale, we use the diagnosis data that's there in the cancer registry from everybody, and we feed some of that back to the regional clinical genetics services. So when you guys go along for genetic counselling, the geneticists can check with us and verify all your family histories so that they can give you the best genetic counselling that's tailored to your situation. So that's broadly how we work. Just to go into a little bit of the science, I'm sure many of you are really familiar with this already. But when we talk about DNA testing for cancer, there are two types, effectively, of DNA testing. There is somatic testing, and that's when we extract DNA from the tumour. And all tumours contain DNA mutations. Whether you've got Lynch syndrome or not, if you have cancer, that tumour contains DNA mutations. A germline test, on the other hand, that's usually done from blood or saliva, and that picks up inherited genetic variants. So if you have a germline genetic variant, then you might have Lynch syndrome, and those are heritable, so the tumour ones are not. But often it's the same genes that we find mutated in the germline and in the tumour. So on this slide to the left, we've got somebody who's got, say, a colorectal cancer with mismatch repair deficiency but don't have Lynch syndrome, and on the right hand side we've got somebody who has Lynch syndrome, so that mutation is in every cell of the body. So this is important for our data sources and the sort of data we collect. So what we are doing in our molecular team, so to put it in context, 6 years ago there was no genetic data in the National Cancer Registry, and I came in at that point, and Jem, the big boss whose picture John showed you earlier, he sat me down on my second day and said, right, your job is to sort out molecular data for cancer and rare diseases. Off you go. And so quite a small remit. So I was a team of one for 3 years, and I made a certain amount of progress, but it was challenging to do this single-handedly. And then about 3 and a bit years ago, Stephen came along as my manager, and he's been amazing, and he's just built up the team around me. So now we've got a really fully functional team. So what we're doing with Lynch is we're collecting the data directly from the NHS labs. So at the top, we've got the germline data, that's the inherited cancer data. And for MMR genes, we've got about 18,000 test results to date, and some of them stretch back sort of 20 years. For the tumour data, when we look at that for MMR genes, we've got data on about 43,000 tumours, and that's stretching back to 2016. So the germline stuff, for technical reasons, our bioinformatician, our IT guys, process that using some really clever algorithms. And our tumour data, our wonderfully talented cancer registration officers record that manually. So we've developed a big genetics education and training programme so we can train up our cancer registration staff to understand and process complex genetic data. These people are not scientists, they're admin grade staff, and what they do is it's just amazing. They do a super specialist job, and we've got them doing some really complex genetics. They're brilliant. So we add all this into the genetics data tables that we've built as part of the cancer registry, and then that goes into the analysis server where we can have a play around, crunch the numbers, and then produce some juicy reports at the end. So that's what we've been doing for the last few years. Just a bit of a disclaimer, none of this analysis has been QA'd. A lot of it's hot off the press literally this week. So what happens in Aston stays in Aston. This has not been published.
Apart from the fact it's going out on the camera.
Apart from the fact it's going out on the camera, but you know.
Don't shoot us.
No, don't shoot us. But actually I feel really privileged to be presenting this to you guys first. So what we're looking at is the NICE guidelines on tumour testing and how they're being implemented across the country, and what sort of proportion of tumours are being given MMR testing. And then I'll talk a bit about our genetic inquiries service. This is all colorectal cancers diagnosed in 2019, which is the first year for which we've got complete cancer registration of all mismatch repair tests, and you can see that this is a postcode lottery. We've split this up by Cancer Alliance, so the best in the bunch are West Yorkshire and Harrogate, who are testing over 70% of colorectal tumours. Remember, every tumour should be getting these tests. Those are the NICE guidelines. Yeah.
Can I just add one thing, Fiona? Sorry to interrupt you, but that point about doing all cancers. We did a really deep dive for the last 5 years in Northumbria Hospital, where we've got really switched on people on all levels, and we only got to 75% tumours tested, because a bunch of tumours, people just arrive already near death. You know, especially an old person comes in with a very advanced cancer. We never get a biopsy. You know, nothing actually happens. They just say this person's got bowel cancer. They do a scan, and they stop. So in that situation, we never get tests. So we probably can never get to 100%, but we could certainly get up to 75, 80%, and it should be higher. The problem, of course, is in this country, so many people are turning up with advanced cancer, you know, as they come in the door, and that's because of the failure of screening. Sorry, York.
Would you not get biopsies post-death then?
Well, no, is the short answer. I mean, you can, but it's just, people don't actually feel the need. It's because, you know, so the hospitals are saying, well, okay, fine, the person's gone, you know.
Would you not be able to identify family members who might be at risk?
Yep, all of that. But remember, this is all cancers. But I'll come back to that at the end, because the fact is, there's a bunch we'll never get to. But they should all be up at 75, 80%.
Quick one. That's England. Have you got the data to embarrass Wales and Scotland and Northern Ireland as well?
No, because we just do England, but we do have a reciprocal agreement with Wales, so it
You
may
might
be
like to ask them to run it.
Don't worry.
We need to talk to Public Health Wales about that, because they we've trained them up as to, and we've given them our genetics data tables and our training, so their staff can now do this, but Public Health Wales own that
Yeah.
data.
Yes, exactly.
I'm just curious, I mean, 16% all the way down the line.
Yeah.
I mean, why such, obviously, you guys all the way at the top in 70%, but 16%, like what's happening in that NHS hospital?
I know, I'm really ashamed, and I was brought up on the Wirral, so Cheshire and Merseyside.
Can we come back? Can we come back to that at the end? Because I think we'll come back to that.
Yeah. This is a real political hot potato, and as John says, we're never going to get to 100%, but that does not excuse.
But if Yorkshire can get to 75, then the Wirral can as well.
Yeah.
But the important point is there is a problem.
Yeah.
So in the work we'll be showing you, one of the things, pushbacks we got was, it's okay, it's all sorted. It's not all
It's not,
sorted.
no.
Now, this might be some failure of reporting here, but we think this is very good data.
Yeah, and if it is a failure of reporting, then the hospitals will have to pull their socks up and report their data better to us, because they have a legal obligation to report their data to us. We can only report on the data that we are given from the hospitals. So if some of these trusts want to come back to us and say, but we've tested
not
many
true.
more than that, we'll
Prove
say,
it.
well, give us the data then. We report on the data we've got. But we now have, this is the first time we've been able to do this. So this is 2019 data. This is the first year for which we've got the cancer registration, and we've been systematically recording all mismatch repair tests. So we had a clue that this was going on, but we've now got the data to back it up. Yeah. Okay. So that was tumour tests. Moving on to germline tests now. This is slightly older data. This is the percentage overall of colorectal and endometrial patients who've had a germline test. It's not very high, but as you can see, the general trend is for it to go up, and we will marry that up with the 2019 data. We just didn't have time to finish that off this week.
It should be about 8 or 9% if we're doing our job properly.
Yeah. So now we're going to look at how the tumour testing and the germline testing marries up. So these are the ways you can have your germline test either before your cancer diagnosis or after your cancer diagnosis, and you can have it either as a full screen, which is for families that the genetic services don't know about, or you can have a targeted test. That's if you're already known to genetic services, they know that you have a mutation, they can test other family members for that mutation. Sorry, guys. So we're not looking at the targeted tests in this analysis, because these are families already known to genetic services. We're not looking at the pre-diagnosis germline tests, because they will have been tested for a good reason as well. The bulk of them, 324 in this year, 78% of all germline tests were the full screen tests post-diagnosis. This is our opportunity to find new cases of Lynch syndrome from tumours. So this is the subset that we're looking at. I'm sorry this is so small, but at the top we've got the number of colorectal cancers diagnosed in 2019. The green bits are whether they had a particular test, the blue bits are the test results, and the pink one at the bottom is whether they had a germline test. So I will zoom into the next slide, and what that is telling you is that overall only 43% of patients had the first-line tumour test. At every stage in that diagram, people are dropping off like flies. So even when they've done the first-line test, they are not following this up properly. And this is why at the bottom we get a tiny number of people having the germline tests. Now there are others having germline tests as well who haven't gone through the pathway quite correctly, and we're in the process of looking at them as well to work out what's going on. Just moving on to finish off the talk now, there'll just be a couple of minutes. This is how we, this is our genetic inquiry service, this is how we support regional clinical genetics services. Back in the day I trained as a genetic counsellor, so that was my first introduction to the National Cancer Registry. So I've sort of seen it from both sides. So every week we respond to about 375 genetic inquiries, so that's over 20,000 cancer family history diagnoses that we can check and verify every year. This is an example of, this is a clinical genetics service in London. Claire Turnbull, Professor Turnbull gave us this case. It was a complex family, so this is a biological brother and sister who had been adopted into different families, and there wasn't a great deal of contact between the two siblings. But the sister had a pancreatic cancer that had been diagnosed at age 59, and she thought her biological brother had had a lung cancer. So she went along for genetic counselling. The genetic service checked with us, checked the diagnosis, and it turned out not to be a lung cancer. In fact, that was a bowel cancer diagnosed at 49. So because of that, the family hit the threshold for MMR germline testing, and those are the criteria. So those criteria were met in this family, and we found they found an MMR mutation in the lady with pancreatic cancer. So because of that, her daughter was eligible for colorectal screening because of her family's diagnosis of Lynch. And at her first colonoscopy, they found a large adenoma with invasive features. So if we think about what we started with, we thought it was a lung cancer and a pancreatic, a pretty unremarkable family history, and in the end we have a family with Lynch syndrome, and probably a life has been saved. So that's the value that we as a National Cancer Registry can add to the clinical genetics services. So, yeah, I think, the conclusions speak for themselves. We're not doing well enough across the NHS. There is a bit of a postcode lottery over Cancer Alliance. Only 43% of colorectal cancers are getting MMR testing in the first place, and even when they are tested, only 44% of those are being followed up with the next test in the pathway. So as I say, we're going to publish these results of Cancer Alliances on Tuesday for Lynch Syndrome Awareness Day, and then we'll get these into a proper scientific journal hopefully later this year with a bigger analysis. And I did some rough calculations, and I think if the NICE guidelines were implemented properly, we could potentially identify up to 700 more people per year with Lynch syndrome with colorectal and another 200 with endometrial. So we could make, the NHS could make a big difference if they were doing it properly. And just a thank you, and a special thank you to the labs who are giving us all this data, and that's been a lot of work for the labs. And special thank you to the patients as well, because at the end of the day, this is all patient data that we're using. So thank you to all of you.
Thanks, Fiona. So, I mean, this is very powerful, as you can see, and very important work, and we're very pleased to be part of it. And again, thanks to Bowel Cancer UK, who kind of gave us the first grant to kick this off, and to Cancer Research UK, who have now kept it going with big money through Claire Turnbull's CanVig programme. So I want to tell you, you may as well sit down for this, Fiona, we can come back to answer questions. This is something we've been working on for 10 years, and we now call it the Newcastle MSI Plus assay. And this slide was made for the what's called the Small Business Research Initiative. For some reason, our hospital is classified here as a small business. It has an annual turnover of £1.3 billion, so it's not that small. But we've got one of these grants, and this came last year, and we're just activating it now. And Kieran, who is the deputy head of our GLH laboratory, he's taken frontline. I'm the academic lead, and Richard, whose work you'll see coming through, one of our former PhD students, now postdoc, is the research scientist on the project. So we've already talked a lot about the MSI immunohistochemistry. In the top left, we've talked about, about staining for the proteins. In the top right is the test for what's called fragment length analysis for microsatellite instability, and I'll show you in a minute what that means. The bottom line is the technology that we've been using by Promega is old-fashioned. It uses fragment length analysis on the old ABI sequences like we used in the last century to do the whole genome project, and now we're doing everything using in the next generation sequences. This test won't work on a next generation sequencer because the fragments are too big. So about 10 years ago, me and my pals got together and said, why can't we work out one of these that will work on a next gen sequencer? You can actually just do whole genome sequencing on tumours, and the bottom right hand shows the abnormal patterns that you see, which can pick up microsatellite instability and mismatch repair, but that's very expensive. Bottom left is what we're looking for, which is abnormal fragment sizes of these repeat segments. So these are the two brains behind the operation. Mauro Santibanez-Coreb is our computational biologist, and Mike Jackson is the molecular geneticist, and they're two senior colleagues. And the three of us have been working together along with Jill and the rest of our team for several years now. So our idea was to create a low-cost, scalable, sensitive, one-step test. So this was a slide put together by Richard. So here are the three options that currently exist. Mismatch repair lost by immunohistochemistry, which actually probably cost about £180. That's slightly over the top. Fragment length analysis costs about the same, and tumour sequencing costs about 500 quid. So what we want to do was slide one in the middle, that we could get this down to about £70, which, when you think that we've got 37,000 colon cancers, these numbers stack up for the NHS. And we wanted high throughput, we wanted it to be automated, and we wanted to put in the other markers, which I'll come to in a minute. So here, this is a microsatellite. So a microsatellite is a really old-fashioned term for back in the days when Ian and I were boys, and we used to use gels, and we used to run things up gels, and used to watch how fast they run up the gel. And things that ran right up to the top were called microsatellites, and they were actually these bits of repetitive DNA. And so repetitive DNA is particularly difficult to copy. If you sort of wrote down a sentence and someone said if you had four or five ands in a row, like one of those things, the distance between Thompson and son is too long when somebody was painting a sign. So if you actually try to copy down all the ands, it's dead easy to get it wrong. You have too many or too few ands. And it's exactly the same for the DNA replication system. And if one of these repeats is in the middle of a gene, it's really serious, because genes are read in sets of 3 letters. If you take one out, everything moves back one, and it becomes nonsense. So that's what we heard about earlier, these frame shift peptides. But what we were interested in were bits of DNA like this, but between the genes, where it didn't really matter, but it was very unstable. And so and this is the summary, basically we can identify microsatellite instability using a panel of these highly sensitive intergenic microsatellites. But because it's on a next-gen sequencing, and because it's PCR, we can chuck a whole bunch of other stuff in as well, especially this so-called BRAF V600E. This is a driver mutation. So it's what you see in about half of the sporadic microsatellite unstable cancers. And in almost all cases, those are not Lynch. So it's really useful to cut down the number of people you have to bring in to do the germline testing. And the important thing is we've now got it down to working on 1 nanogram of DNA, so just a little tiny fragment of the tumour. So the original plan was done by 2 PhD students, Lisa and Ganim, and this is Mauro's idea. We looked for the repeats that were actually linked to a polymorphism, where that's like a blood group, just the difference between the two sides. And that allowed us, using a fancy thing called molecular inversion probes, where we could barcode, so we could count every single reaction. And we did that on a MySeq, which is a bog standard machine in all the labs. And on the right here, you can see that stutter peak, as we call it, could be just the result of the amplification process, or it could be the result of there having been a real slippage in one of the copies. Now, if it was a real slippage, it would be on one or other strand. So it would either be travelling with the A, the red line, or the T, the green line. So in other words, if that stutter peak was all one or all the other, then it's probably real. Whereas if it was equally the two, it's probably just an artifact of the technology. This is important because the old Promega test, you have to have an expert eye to differentiate these stutter peaks from a real shift. We got rid of that. And so these are, Lee was one of our students who's now working in the lab, and Helen's his supervisor. And on the top right-hand corner, on the top of that are the microsatellite unstable cancers, and below that are the microsatellite stable cancers. Now, Ian shouted on a previous seeing this slide, nothing's ever 100% sensitive and specific. So these were known MSI high tumours. So we were basically saying, can we always see whether it's unstable or not? If you look at the old reports, they sometimes say it's microsatellite unstable low, or MSI low, or MSI high. That's purely an artifact of the old technology. We've just binned that now. It's either microsatellite unstable or it's stable. And it's a clear, and you score all the markers and you get this cost. So we're very simple to use. Lizzie is a fantastic bioinformatician in Sheffield who's part of our genome laboratory hub. And she set off to make an MSI app for the lab. So basically all this data drops into the app, and here's just one of the screens that the scientists see. On the left, you can see that little dot below the line showing that this was definitely a stable tumour. And on the right, you can see that it was BRAF negative, that's the top line. But we've also chucked in all the other driver mutations that you might be interested in. So the oncologists like to know about the RAS mutations because they'll tell you whether the drugs work or not. So we can basically chuck in a whole bunch of other stuff and you get the whole report in one go. And so these can now be turned around in 10 days in our lab, but with our new technologist who starts on Monday with our new grant from the government, that will go down to under 7 days. And we've now been given funding to roll this out through this SBRI award to all of the country. So we will pay for a full-time technologist in each of the genome laboratory hubs, along with enough money to run this assay at speed and scale, and at least 1,000 tumour content analyses. And that will hopefully get this embedded, because it's now, the other thing is, from the hospital's point of view, it's cheap as chips because they don't have to pay for it. It comes out of the national budget for the test directory. So from 2023 onwards, this can be just rolled out as an additional test where you're doing IHC, or instead of IHC where that isn't technically feasible. And so this is just some, I actually made this for the people at NHS England, but I'm not, this is Christine, our technician, actually cutting these sections off blocks. It's a very simple process, and essentially what you end up with is what are called curls. So we can actually do this analysis on one curl, 10 microns thick. So that's in the forceps there. So essentially it works on a tiny, tiny amount of DNA. That's important because what we were finding was very often these blocks, you don't get very much DNA from it because basically my pathological friends like Ian just kind of beat up the tumour with all of their fixation and stuff. But no, so basically the DNA gets mashed up in the process of fixation. We can improve on that, but if you can do it on 1 nanogram of DNA, then you'll get a result every time. Whereas sequencing the genes often doesn't work. And so these are the blocks from 4 patients with Lynch syndrome in our local hospital. And what we did here was to just test the question of what if we just took a slice of every block and tested for microsatellite instability. But also, we'll test each individual biopsy because our pathology colleagues say, well, we're supposed to do tumour content analysis. And so we said, well, okay, if you want to, but you don't really need to. And so these are the results of that. So basically on the right-hand side are the individual biopsies, and sure enough, 2 or 3 of them missed the tumour. In fact, 5 of them missed the tumour according to the microscope, but we still got an MSI result because, of course, you get microsatellite unstable tissue around the tumour. The black dots in the middle are the important ones. Those are where you've just taken a slice across the whole block and it comes up MSI high every time. So we're now going to validate this, but basically this takes a whole slot out of the process. If we can just take a slice of every colon biopsy block and run it through this test, it takes a day off the turnaround because we don't have to wait for somebody to do the tumour content analysis, which costs £49. I know because I have to pay for it. The other even more exciting thing is that the team are going onward and upward. No, I'm very sorry. And so this is Rachel, and Rachel, where is that? Has been working with Mike on some even more sensitive markers, very cleverly derived from people with, remember you heard earlier about constitutional mismatch repair deficiency. So these are children who get it from both parents and survive, and they're very, very rare, only about 1 in a million kids will present with this. But they have got such sensitive systems that they get microsatellite instability in normal white cells in their blood. And so what we did was we did whole genome sequencing on those kids to find the markers that were consistently unstable to give us even more sensitive markers. And that's what this panel is showing. So along the bottom is the set we were originally showing with a little gap between the stable on the left and the unstable on the right. With our new markers, the stability separation is, the stable versus unstable is massive. So this is very exciting. So that's allowing us to improve the assay and allow it to work even faster on even smaller amounts of DNA. But it also gave me an idea. I thought, if it's this sensitive, maybe it'll work in other places. So we said, let's try and get some urine from somebody with a urothelial cancer. And this is a presentation I did for Rachel, because just before she was about to present this at the National Cancer Research thing, she gave birth to Amelia, and so who was the youngest member of our research team, and so I presented it on her behalf. And so basically we've already discussed this. I quite like this, couldn't it? So we've got 4 genes, but they're 4 very different patterns, and in MSH2 in particular, 18.3% of people with MSH2 will get a tumour in their urothelial tract, the kidney, the ureters or the bladder, and about 10% of them will get in the bladder. So that's a lot. But we don't offer any effective screening. We do sometimes look at the urine, but it's completely useless, as we've demonstrated in the past. In 2018, 10,243 urothelial cancers were recorded by Fiona and her team. 43 of them, as far as we can tell, had a germline test for Lynch syndrome. So we're basically completely blindsided on this problem. We have no surveillance program. So the question is, could we test the urine of somebody? And Alan, who has allowed me to show his picture, presented in our CAP study with the urothelial cancer. So we collected his urine, and look what happened. That's Alan up on the top right there, just after I'd abused him by taking a blood sample from him. So on the left are the markers, and you can see there's the variant allele frequency is looking for those funny little peaks. So you can see in his blood, there's nothing there. But in multiple urine samples, the next column along, this column here, you can see there's lots and lots of these markers are unstable, very similar to the markers in the tumour. And in his urine, about 70% of the DNA was from his tumour. As soon as we took his tumour out, his markers disappeared. So we fixed it. And here's just the red one there, just to show you the peaks. I mean, this isn't hard to see. It's barn door abnormal, passing horseman stuff. And so essentially, this potentially is now a screening test for urine. And we've got Rebecca Hall, one of our genetic trainees, is just starting this month as for a year's project to see if we can do this with Rakesh here in urology, to start checking urine from people presenting with renal cancers and to reach out. So the first thing is, if anyone in your families or anyone you know has a renal cancer with Lynch syndrome, give us a call, because we'd really like to run this test to see if it shows. And what we now need to do is demonstrate its sensitivity and specificity, as you heard from Tristan. We know it works in Alan. The question is, will it work consistently, and how reliable will it be? I then got this email, and this is my last but one slide. Now, this is not Boris Johnson's reply to the Metropolitan Police. This, in fact, is an email that I got in November from a lady we'll call Anne. And Anne is a scientist who works for the government. And she actually used her work email to send me this message, and I'll just pick out 2 things. My dad was seen by you and it was identified he had Lynch syndrome after having bowel cancer 3 times. I have the gene. I have issues with my womb. I had a scan, it was identified that I have a fibroid or polyp. I've been waiting for over 2 months for a biopsy, and it says in the text for over 6 months since I first identified. And in fact, she presented in October of 2020 with severe menstrual bleeding. You heard earlier in the talk. She's got an MLS1 mutation, 40% chance of endometrial cancer, and a year on, we're still discussing what to do, and she's being reassured she's got a fibroid, she's 38, it's probably just a turn of the leg. So I discussed our new assay with her, and she said, yeah, let's give it a go. So she'd arranged anyway to see one of our gynecologists privately, so we got a vaginal swab and a urine sample, and here's what we saw. On the left was the mouth swab showing the normal pattern. There's the, oh damn it, go away. And there is the tumour, a coliP urine, a Shrek urine, these are 2 different preservatives, and the vaginal swab. Absolutely barn door. Now, Emma Crosby last year published that you could look down a microscope and see endometrial cells, cancer cells in the urine. But this, of course, is much, much easier. These will last 7 days in the post. You can collect the samples, and we could potentially start MSI screening urine for both endometrial and urothelial cancers. Again, this is just the first step. This is the first time anybody's seen this. We won't be actually doing the post-op urine test until Monday. But she actually had a high-grade endometrial cancer, which would probably have done for her had she been left waiting yet more months for a routine hysterectomy. So, in summary, aspirin cuts the cancer rate in people with Lynch syndrome. It's safe in most people. Side effect-wise steeply after the age of 70. Resistant starch, fermentable fibre reduces the number of non-CRCs in Lynch syndrome. We need the DNA spell checkers in our cells to correct errors when cells divide, losing both copies cause problems in repeat segments. We can see the mismatch repair system isn't working if the proteins are missing or there are lots of errors in repeat segments. We still aren't doing enough screening in CRCs. MSI plus can help. We are doing better in endometrial cancers, but it's still too slow. We can pick up MSI high DNA in urine. We need your help to prove it. Thank you.